Hi, I'm Ryan Sullivan, and today I'm going to be presenting on our ARDC Platforms project, the Australian Imaging Service. Nationally, institutions and researchers alike have been challenged with handling sensitive data. We are working to build a national platform to address this challenge for imaging data to better allow high-end analysis and collaboration across the country. We do this by integrating with clinical sites, including imaging instruments, PACS, VNAs, and clinical viewers. And importantly, we are able to have consistent de-identification and secure data transfer and management on the platform, having controlled access and auditing capabilities of said access. The entire platform is browser accessible, allowing researchers and clinicians alike to view and annotate their data without having to install any local software. Furthermore, we are able to expose high-powered computational capabilities and analysis pipelines directly in the browser, allowing easy automation and one-click pipelines and reports to be generated without the user ever having to see a command line or terminal interface. Neuroimaging is arguably the most mature computationally of the imaging fields, in part due to the high replication of the target sample, i.e. the human brain. I will point out two impressive projects that are being presented during this conference, particularly Neurodesk and the Australian Characterization Commons at Scale, as two Australian exemplars in this area. The issue, however, is that neuroimaging is not all imaging, and there are many other types, and many are insular and don't always talk to one another. I'll split the different types of imaging along three axes. So the first is looking at sensitivity. Traditionally, researchers collect data, they do their analysis, they publish, and then they share the data. However, sensitive data, particularly cultural or of medical nature, cannot as easily be shared, and there are challenges around collaboration and analysis. On the other extreme, in the GLAM sector, there are types of imaging where researchers are not just looking to quantitatively analyze the images, but also have qualitative visualization for training the next generation of researchers, of clinicians, and for engaging with the public. These include things such as anatomical images, ancient artifacts, and ecological samples, both of animals and plants. On the second axis is different types of imaging. MRI is usually used for neuroscience, but there are many other modalities such as CT, ultrasound, PET, and nuclear medicine, dermatology, photography, etc. And there are, of course, the different fields of research: physiology, vet science, cardiology, and the list goes on. So what we are aiming to do is, with our platform, to bring all of these together in a single place and start adopting common tools that can be used across. In particular, three of the exemplars on which we are focusing are dermatology for sensitive full body photography, a generalizable MRI raw instrument data, which converts all proprietary to an open format, as well as electrophysiology for neuroscience. Now, how do we want to get there? Over 2 years, we have proposed 5 streams of work with 4 community outcomes. The first stream of work is operations. This is looking at how do we have consistent ethics and de-identification so that you can run multi-site trials across different states. Looking at implementing the trusted data repository framework that was previously developed by the NIF and AIS project, as well as looking at the long-term sustainability. The first development stream is deployment, which is looking at leveraging containerization to make it easier to deploy a core node, as well as to deploy the edge devices that sit next to the instrument or in the clinical site. The second technical stream is pipelines, again using containerization, looking at three broad categories, particularly quality assurance, open format conversions, and analysis tools and toolkits. The third development stream is looking at expanding the modalities. So this goes back to trying to encompass as many imaging modalities and associated data types that may occur during an imaging session into the platform. And lastly, no platform should stand on its own. So we are looking at integrating with other common technologies, including RedCap, NVIDIA's Clara for GPU accelerated research, and Jupyter Notebook. We foresee the outcomes of these projects to also result in 4 community pillars. The first is just a community of practice around imaging infrastructure. The second is a findable project search. A lot of the data that we deal with is sensitive and needs to be protected, but that makes it difficult to find. So we will have a front end that houses project level metadata that points to the secure data repositories. Thirdly, we will have a trusted tool repository. A lot of work has been done on trusted data. We think it makes sense to, in the next phase, look at how do we do trusted analysis of said trusted data. And lastly, looking at a national compliance statement so that every node of the Federation will meet a minimum standard in terms of security, capability, user experience, and ethics and data identification. So what does our Federation look like? In blue, I've listed the number of AIS nodes that are part of the current 2-year project. And in pink, I've listed a number of clinical sites which are already integrated or which we are working with to integrate as part of this project over the next year. Additionally, we are looking internationally as much research, particularly human research, needs to be done internationally to not have selection bias. We have a number of sites labeled in pink, which are currently or will soon be sending de-identified data from international countries to one of the Australian nodes. And we furthermore have 2 potential nodes, one in the US and one in the Eurozone, as part of a Medical Research Future Fund program. So how are we actually doing this? First stream is deployment and configuration. We are looking at how to consistently deploy XNAT with Australian Access Federation, which includes a number of viewers, searchable mechanics, an extensible API, as well as implementing edge devices at university and clinical sites that can both de-identify and upload the data to the target node. The second stream is really about building the computational capability. So XNAT by default has a container engine for running pipelines. We really want to build out that library, but importantly, build out that library in a trusted way so that for certain use cases, we have pipelines that are approved by the national imaging facilities for that use case to try to increase that reproducibility in science. And the third stream really goes to expanding those modalities. So this is looking at converting from proprietary formats to open standards, particularly using the NIH's Gadgetron for MRI case space, as well as changing our upload tools to be able to capture a wider variety of data that is produced during or adjacent to an imaging session and makes sense to really view alongside the imaging data for interpreting the results. And lastly, we look at integrations. This is particularly looking at around NVIDIA's Clara for GPU accelerated pipelines, as well as RedCap, secure VMs, the characterization virtual lab, and importantly, multiple ARDC platform bids for the 2020 scheme, should they be funded. So what is the type of thing that we ultimately want to support with this platform? I'll take the ACEMed project as an exemplar. So the ACRF Center of Excellence in Melanoma and Dermatology has a national project to deploy 15 full-body dermatology imaging devices across the east coast of Australia. So 5 in Victoria, 5 in Queensland, and 5 in New South Wales. We are working with the ACEMed project to build their research repository on the AIS platform. In particular, building it on the UQ, the UCID, and the Monash nodes. This will cover roughly 25 patients per week, per site, over 15 sites over 3 years, with the first 3 sites aiming to go live in Q1 of next year, along with integrating with secure computational environments at each of our institutions as they develop. I'd like to quickly go over the technologies that I've mentioned. So XNAT as the core technology, the Open Health Imaging Foundation Viewer, RedCap, Gadgetron for MRI, Clara for GPU, Jupyter Notebook, as a more integrated approach for this deployment, as well as Docker and Kubernetes, and then working with Radiologics, which is a private vendor for XNAT support and custom development, Palsy, QSIF, and NVIDIA. We have a number of academic partners as listed here, and we always welcome more. We work with a number of clinical partners, both public health across the 3 states, as well as private clinical vendors, with the view of trying to really support everyone that's doing research. And lastly, here is the team that is working to make this happen. We have a core development team, along with an academic and technical oversight group that guide our work. And we, of course, can't do this without the help of the XNAT Energy team, Radiologics, and countless research groups to guide our direction. Funding is provided by the ARDC for this project, by the ACRF for the ACEMed project, and by the Department of Health for the Medical Research Future Funds component for the international deployments. Now, happy to take any questions.